Welcome to the Sainsbury Laboratory in Norwich. This is the first video in a series about the Golden Gate cloning DNA assembly method. In this video, we'll learn about type 2S restriction enzymes and how they differ from classical type 2 restriction enzymes. Restriction enzymes cut DNA by making two incisions, one in each sugar phosphate backbone. They are classified into types according to their structure and whether they cut their DNA substrate at their recognition site, or if the recognition and cleavage sites are separate from one another. Classical type 2 restriction enzymes are commonly used in molecular cloning. Each enzyme recognizes a specific sequence, usually palindromic, shown in orange. They cut their substrate within their recognition sites, leaving either sticky, that is 5' or 3' overhangs, or blunt ends. When a desired fragment of DNA, shown in blue, is released from a plasmid using a pair of classical type 2 restriction enzymes, part of the recognition sequence, shown in orange, will be present at the ends of the released fragment. This means it can only be ligated to other pieces of DNA that have compatible ends, usually those that have been cut with the same enzyme. When this happens, restriction enzyme recognition sequences will be present between the two fragments. These are known as assembly scars. Golden Gate cloning uses type 2S restriction enzymes which cleave outside of their recognition sequence. For example, the recognition sequence for BPI1 is GAA-GAC, shown in orange, but the cut is made in the 6 base pairs downstream of the recognition sequence, leaving 4 base pair 5' overhangs. There is no sequence requirement for the identity of bases in the cleavage site. In this example, a fragment of DNA, shown in blue, is flanked by two BPI1 recognition sequences, GAA-GAC, in opposing directions, shown in orange. When this fragment of DNA is released from a plasmid using these enzymes, no part of the recognition sequence will be present in the released fragment. It can be ligated to other fragments of DNA that have compatible ends, and, when the two pieces of DNA are joined together using a DNA ligase, no part of the restriction enzyme recognition sequences will be present. This is the end of the first video. In the next video, we'll talk about how type 2S restriction enzymes are used in Golden Gate cloning.